Okay, so welcome back to this presentations on amino acids. And here we will be discussing on the classifications of amino acids. On the basis of general chemical characteristics of their side chain, the amino acids can be divided into these many. The first one, hydrophobic amino acid with non-polar R group, polar amino acid, positively charged and negatively charged amino acid. Okay, we can just check it once, one by one in detail. Non-polar aliphatic R groups. Okay, that is when the side chain is non-polar. What is non-polar? Non-polar is actually hydrophobic in nature. Okay, and it is aliphatic. I hope you know what is aliphatic and aromatic from your chemistry classes. Okay, so the side chains here are non-polar and hydrophobic, and the major amino acids that are that can be listed under non-polar aliphatic groups include alanine, valine, leucine, isoleucine, glycine, methionine, and proline. Okay, we can go for like gavlip, fine, and methionine. Methionine, obviously, you know that it is the during the like translation process, that is the first uh, the amino acid complementary to the start codon, isn't it? Yeah. So coming back here, the alanine, valine, leucine, isoleucine, they tend to cluster together within the proteins. When proteins form the secondary, tertiary structures and all, the R group, which is aliphatic or which is hydrophobic in nature, usually they remain try to remain inside the protein cluster. Okay, why so? So that it can ward off interaction with the water or aqueous medium outside. Okay, so it to stabilize the protein structure, it actually starts to. So if this is the okay, we'll see. Right. So if this is a protein structure, okay, fine. So you have plenty of amino acids. One, two. Okay, just 1 minute. 1 minute. 1 minute. So if we have plenty of amino acids and we are okay. So just imagine we have a protein chain like this. Okay, sorry, amino acid chain like this forming a protein or polypeptide. Okay, we have one amino acid over here, second one here, third one here, fourth one here, likewise. Okay, so when there is a hydrophobic amino acid, okay, that is an amino acid with hydrophobic side chain group. Okay, just imagine this is the one. Okay, here what happens is all around the protein, it could be cytoplasm or any other biological medium which contains more of water, almost 75% water it is, isn't it? So what happens? The R group will tend to extend into the central core of the protein. Okay, into the central core. Fine. Why? So that it can avoid interaction with the water here. Okay, so one thing. And secondly, to stabilize the protein structure, this what you call the R chain, which is hydrophobic, it develops hydrophobic interactions with other such groups within the protein core. Okay, so this is what happens. I hope it is clear. Okay, so we have the R chains of this particular group, it being the non-polar and hydrophobic. Now we can see glycine, it is the simplest structure with actually what you call, it is having the R group, simple H hydrogen. Okay, so it is the simplest structure we can have. You can see here glycine. Okay, glycine, it is R. This boxed ones, it shows the R group. Okay, here it is the simplest one with hydrogen as the side chain. Okay, so this is the simplest. You can see the what you call amino acid, and it is very slightly hydrophobic in nature. Okay, next we have is the methionine. Methionine is a sulfur containing amino acid. You can see here it is sulfur containing amino acid, and this is this has slightly non-polar nature. Okay, so this thioether group, SCA, this group, it is actually slightly what you call non-polar or hydrophobic in nature. Proline, on the other hand, you can see the structure. It has an aliphatic side chain with distinctive cyclic structure, and it is known as an amino group. Okay, you can see it is amino group of proline. Okay, here you can see the proline. It is, this is what is known as the amino group. Okay, instead of NH3 plus, it is having NH2 plus, isn't it? So this forms an amino group. Okay, it is this actually proline is usually found in those parts of the proteins where there is a bend. Okay, wherever there is a proline in the amino acid chain, so at that particular point there will be a bend. We call it as a kink. Okay, that's a bend, right? And you can see how this what you call the hydrophobicity it increases. So proline, you can see uh, Gavli. Okay, so it is hydrophobicity increases in this way. Okay, uh, you can see from glycine you have simple hydrogen, then alanine methyl group, right? Then valine, you can see it is CH, CH3 twice, and leucine and isoleucine, it is almost like C4H. What is it? Yeah, CH2, CH, CH3 twice. Okay, almost the similar way here. Okay, so you can see the hydrophobicity increases. Okay, as many carbon chain as it is increasing in length, the hydrophobicity also increases. Okay, so this is about the first group, that is non-polar aliphatic group. The second is the aromatic group. Aromatic group, I hope you know, it may contain benzene or phenyl chain, isn't it? Ring, actually. And there are three groups: phenylalanine, tyrosine, and tryptophan. They are all aromatic compounds. And they are hydrophobic in nature as well. All these can participate in hydrophobic interactions because of their nonpolar nature. The tyrosine, you can see it contains a hydroxyl group. Okay, and this hydroxyl group, it can form hydrogen bonds. So it is a very important functional group in some enzymes. Then tyrosine and you can see the tryptophan, they are more polar than phenylalanine. Okay, so this is more what you call hydrophobic in nature. Now you can see, so these can form interactions, what you call hydrogen bonds. So it can very well interact with water molecules. So it is less hydrophobic as compared with phenylalanine. Okay, so these are the aromatic compounds, which are the phenylalanine, tyrosine, and tryptophan. Okay. Then we have is the polar uncharged groups. Uncharged in the sense they are not what you call hydrophilic or like acidic or basic in the sense. Okay, so here the R groups are more soluble in water, that is it is hydrophilic in nature than those of the non-polar amino acids, and these contain functional groups that can form hydrogen bonds with water. You can see hydroxyl group over here in serine, right? 309 also contains a hydroxyl group, right? Cysteine, this is cysteine. Cysteine can form hydrogen bonds with, or actually it is not purely hydrogen bonds, it is that is a disulfide bonds with another cysteine molecule, forming a compound known as cysteine. Okay, so it is the cysteine molecules which can form disulfide bonds. Then asparagine, you can see here double bond O. This O oxygen can form hydrogen bonds with another molecule. Okay. Then here again C double bond O. You can see here these can form the hydrogen bonds with. Okay. So next one we have is a positively charged R group. Positively charged in the sense, usually we have NH3 plus and COO minus in a particular amino acid when it is in a biological medium. But if it is having another NH3 plus or an amino group, obviously it becomes more positively charged. That is, when there is an extra NH2 plus, it is more basic also, isn't it? That is what positively charged basic R groups. So the most hydrophilic R groups are those that are either positively charged or negatively charged, and here it is positively charged because of the presence of an additional NH2 group. Okay, so here you can see there is an NH3 plus here, this two amino groups over here, and the histidine here. Okay, so this makes it more basic in nature, and it can also get into hydrophilic interactions. Okay, um, three examples: lysine, arginine, and histidine. Okay, now next one is negatively charged. As always, it may contain an additional CO carboxyl group. Okay, apart from the carboxyl group which is attached to the alpha carbon, there will be an additional carboxyl group, and that makes it more of negatively charged. Two groups, that is aspartic acid and glutamic acid. Okay, two acidic forms. aspartic acid and glutamic acid. There are complementary amide forms, that is aspartame and glutamine is also found. Okay. So this is regarding the what you call classification based on the chemical nature of the R group. Okay. Okay, so now next kind of classification is based on the structure again. The first one, it is amino acids with aliphatic side chains, as we have seen in the previous classification, where it is monoamino monocarboxylic acid. That is, there is only one amino group which is attached to the alpha carbon, and one carboxylic group which is attached to the alpha carbon. Okay, they are aliphatic in nature, so it is obviously hydrophobic. Okay, all the aliphatic mode of aliphatic chain, more hydrophobic it is. Okay, so it includes glycine, alanine, valine, leucine, and isoleucine. The second one is hydroxyl containing. Okay, it is serine, threonine, and tyrosine, which contains hydroxyl group. When there is a hydroxyl group, obviously it can take up hydrogen bonding. So it is hydrophilic in nature. Sulfur containing compounds, mainly two, that is cysteine and methionine. Cysteine, it do have sulfhydryl group, SH group, and methionine thioether group. Okay, SCH3, that is that you will have C grouping. Okay, you may have seen it in the previous slides. Okay, so that is methionine, and these two can form SS bonds, the disulfide bonds. Okay, then acidic amino acids and their amides. Acidic amino acids, I needn't tell you that it is apart from the carboxyl group attached to the alpha carbon, there will be additional as carboxylic acid. Okay, so it is dicarboxylic acid, monoamino acids. Okay, so amino acids having a single amino group and two carboxylic group. So two examples are we have only two, that is aspartic acid and glutamic acid. And then there are amides. Okay, that is asparagine and glutamine. These are the amides of these what we call aspartic acid and glutamic acid. They are amide derivatives of the acidic. Then we have is the basic amino acids. Basic amino acids, it is dibasic monocarboxylic acids. That is, there is only one carboxylic acid group and two amino groups are there. Okay, so it is dibasic monocarboxylic acid acids, and we have three, that is lysine, arginine, and histidine. Okay. And then we have the aromatic amino acids. Obviously, we have already seen it is phenylalanine, tyrosine, and tryptophan. Okay, tyrosine is also hydroxyl containing compound, but it is major part is actually the aromatic compound. Okay, and the last one, it is amino acid, proline group, where it is not NH3, instead the NH3 is, exists in the form of amino. Okay, it is amino. It is an amino acid, proline, because it contains, instead of NH3 plus, it contains amino group. Okay, so this is one set of classification we have. Another is based on the polarity. Obviously, all these classifications, it is based upon the nature of the R group. Okay, so based upon the polarity of the R group, we have the first one, non-polar amino acids. Non-polar in the sense they are hydrophobic. Okay, they don't have any charge on the R group. Okay, so basically it is dipolar in nature. It may be like just because of the carboxylic acid and amino group attached to the alpha carbon, the charge may exist. Otherwise, R group, it is neutral in nature. Okay, so it is alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan, and proline. Okay, how does the R group attains charge? Either it should have a amino group or it should have a carboxyl group. Okay, so if it doesn't have any of these, R group doesn't have any additional amino group or a carboxyl group, obviously it is non-polar. It is grouped under non-polar group. Okay, the second is polar amino acids, that with no charge on R group. So it is what you call polar in the sense it is hydrophilic. It can form hydrogen bonds with what you call other molecules. Okay, they are hydrophilic in nature, right? Polar molecules. So they possess groups like hydroxyl or sulfhydryl SH, amide, NH, and participate in hydrogen bonding. Okay, so it includes glycine, serine, threonine, cysteine, glutamine, asparagine, tyrosine, etc. The next we have is the polar amino acids with positive R group. Okay, so it is also polar in nature, that is what you call, it is hydrophilic in nature, but unlike the previous one, it is positively charged R group. Okay, it includes lysine, arginine, and histidine. Okay, then we have is the polar amino acid with negative R group. Okay, so it may contain an additional carboxylic acid. Okay, so it is aspartic acid and glutamic acid. So this is about the classification based on the polarity. Next one is based upon the nutritional classification. Essential, non-essential and semi-essential. Okay, we can continue the next presentation.